Hello guys and welcome back to Excelsior Academy. So in this cell walls and the extracellular matrix, the cell interactions, we have completed about the cell walls and we were talking about the extracellular matrix, right? Now proceeding with that, so let's talk about the adhesion proteins. So already about the adhesion proteins we have discussed, like how it is helping in making connections between the two and also with the extracellular matrix. Okay, so let's see. So adhesion proteins, they are the third class of extracellular matrix constituents which are responsible for linking the components of the matrix to one another and to the surface of the cells. So what does it mean is, so you have got the two cells. So it is making connections here and it is also making connections here. Okay. So those are your adhesion proteins. They interact with collagen and proteoglycans to specify matrix organization and are the major binding sites for the cell surface receptors such as integrins. Okay, so these integrins are coming under your adhesion proteins, which is helping in making the interactions between these cells. The prototype of these molecule is fibronectin, which is of the principal adhesion protein of the connective tissue. So in the case of the connective tissue, the main adhesion protein is fibronectin. So it's a dimeric glycoprotein which consists of two polypeptide chain and each consists of around 2500 amino acids. Within the extracellular matrix, fibronectin, it is often cross-linked into the fibrils. Now, fibronectin will again have binding sites for both collagen as well as the proteoglycan. So it cross-links these matrix components. Now, what is happening is that, so as you know, you have got your collagen, right? So they are present in the sheet. So it will be present like this somewhere, SA in the extracellular matrix. Then in between you have got your proteoglycans like this. So if you remember proteoglycans, they are having the protein core, and to the protein core your disaccharides are added like contractin sulfate, dermin sulfate, keratin sulfate, right? So those are added. So they are present over here. Again, they are the negatively charged one. Okay. Now, in between of this, fibronectin will be present here like this, making the cross-linking. Okay, so a distant site on the fibronectin molecule is recognized by the integrins. So what we are saying is that this fibronectin, wherever it is present, it will be recognized by the integrins. Or integrins ka kya kaam hai, if you remember? Yes, Akshaya, would you like to try what integrins does? No? Do you remember like when we were talking about the cell surface, the plasma membrane, then we have got the integrins, and with the help of the many other proteins, it is bound to your actin filaments. SSSA. The fibronectin is making connections with the other cell having the other proteins over here. Okay, no issues. Now look at over here. So this is like showing how there is a interaction between the cell and the extracellular matrix. Okay. Now look at in the figure. So as you can see, we have got the actin filaments are there. Sorry. Actin filaments are there. Then so many complex proteins are present. If you remember, we have discussed all about this complex proteins which are present, plakophilin, plakoglobin, all these kinds of different types of proteins which is present during the interactions. We have discussed all this thing. So right now it is the focal adhesion proteins. Now they are attached to the integrin molecules. Okay. Now integrin molecules are then attached to your fibronectin. Can you see? There is this attachment with your fibronectin molecules. Now, fibronectin molecules and integrin molecules are in a connection with the different proteins. Now, fibronectin is also making connections with the collagen, where collagen is cross-links with your proteoglycans. Okay, so everything is in attachment, like it is like it is like making connections with each. Now, the question is coming: if if these the cell is there, okay, the cell is there, and here you have got the extracellular matrix. Okay. If suppose the cell wants to move, but it is all surrounded by this extracellular matrix, then how it is supposed to move? Like it is gonna move in between of these collagen fiber, or kaise? Like what will be the best way in order to move from one place to another? Shruti? Like how the cells are able to move when they are bound to these extracellular matrix? Any guesses? Akshaya, Shruti?
Maybe they can dissolve the connections and remake them.
Okay, and how are they going to dissolve the connections?
Maybe using the same method that they used to originally dissolve and rejoin the connections when they are made.
So not exactly, but this is correct that they try to remove all those connections. So how they're going to do is that with the help of the enzymes. Okay, so what they do is the moment they get a signal that the cell has to move in that particular direction, they start releasing the enzymes. Of course, it is going to be again the extracellular enzymes. Okay. Now, I need to break all the connections with the collagen and the others. So I'm gonna release enzymes like collagenase. Okay. So it will start degrading the collagen which is present on the path of that particular cell, and the area will be cleared so that they can move ahead. Okay. And these collagenase, they come under the category of metalloproteases. They are coming under the category of metalloproteases. Please remember this. Especially they bind to zinc 2 plus. Okay. So the principal components of the basal lamina are distance adhesion proteins of the laminin family. थोड़ा सा ध्यान दो. This is laminin family, and earlier we have discussed about the lamins, right? These are your intermediate filaments which is present below the nuclear envelope, okay, or which helps in forming the nuclear envelope. So those are lamins. Here it is laminin, okay, which comes under the adhesion proteins. Now, laminins are cross or T-shaped heterotrimers of alpha, beta, gamma subunits. They self-assemble into mesh-like networks. The different laminin subunits also have binding sites for cell surface receptors. Like, it can bind to your integrins, it can bind to your proteoglycans. Now, they are tightly associated with another adhesion protein called nidogen, which binds to type 4 collagen. ये भी important है. This slide is important, please. Huh? Okay. As a result of these multiple interactions, laminin, nidogen, collagen, and the proteoglycans form cross-link networks within the basal lamina. So what is basal lamina? It is nothing but the cross connections or the cross-link networks of the adhesion proteins with your collagen as well as your proteoglycans. ठीक है? अभी हमने क्या देखा? That collagen is present like this in between the proteoglycans are present. Now they all are connected to your laminins proteins as well as nidogen proteins. Okay. Now nidogen makes connection with your collagen, type 4 collagen. Okay. So this is how everything is getting cross-linked. Now this is your laminins. If you see, so it looks like a T shape as they mentioned. It has got your alpha chain is there. Okay, this pink color thing is the alpha chain. Then you have got the beta chain and gamma chain. So as you can see, beta and gamma chains are like, coil. Now, here, this portion is your cell binding, where it is going to bind to the cell. This portion is proteoglycan binding, right? Now, gamma, it has got the nidogen binding. Okay. So laminins make interactions with the nidogen. Nidogen make interactions with your collagen. Fourth type. Okay, as well as the interactions with the proteoglycan. So this laminins is making cross network with the collagen as well as the proteoglycans present in the matrix. Clear, guys? Is this figure is clear? And now this is showing the assembly of the laminins into a network. Okay, so this makes sure that your extracellular matrix is intact. Clear? Able to follow, both of you? Akshay, Shruti? Any doubts in this?
No.
Okay. Now coming to the cell matrix interactions. Okay. So those were the addition proteins which helps in the making of the interactions. Now let's talk about those interactions like between the cell and the matrix. So the major cell surface receptor responsible for the attachment of the cells to the extracellular matrix are the integrins. Okay. Now the integrins, they belong to a transmembrane proteins. They have got two subunits, alpha and beta, right? And they are made up of 18 alpha subunits and 8 beta subunits together. Around 24 different integrins are there. Okay. Now the integrins bind to short amino acid sequence, present multiple components of the extracellular matrix, including collagen, fibronectin, laminin, and the proteoglycans. So integrins क्या कर रहा है? See, from the inside of the cell, okay, here inside of the cell cytoplasm है और यहाँ पे outside of the cell मतलब extracellular matrix is there. So this integrins is making so many connections with the extracellular matrix, like it is making connections with the collagen, fibronectin, laminin, proteoglycans, and everything. So it is like, you know, you need to understand that this is like a cell way of knowing कि बाहर क्या क्या है, ठीक है? In addition to the attaching the cells to the extracellular matrix, the integrin serves as anchors for the cytoskeleton. So they are also in connections with the actin filaments. The resulting linkage of the cytoskeleton to the extracellular matrix is responsible for the stability of cells matrix junctions. And there are two ways by which the cell matrix junctions are formed, that is focal adhesions and the hemidesmosomes. And this already we have discussed in the earlier lecture, right? So focal adhesion and hemidesmosomes. Focal adhesions क्या था आपका? If you remember the movement of a cell, something like this we have made, right? And it starts making the connections. So whenever it is moving ahead, यहाँ के टूटते हैं और आगे के और बनते हैं, ऐसे, 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 right? Okay. So same thing again, same figure, like what all proteins are involved in focal adhesions and hemidesmosomes. So focal adhesion, when the cell is moving and it is making interactions with the extracellular matrix or, you know, on the other like endothelial cells if it is present or wherever it is moving. Now hemidesmosome is interactions of your cell with the, again, the extracellular matrix, which is your basal lamina. Okay, जहाँ पे आपका cell move नहीं हो रहा है, but it is anchored over there. Okay, it is present over there. Okay, so now when cell is moving, so as this we have already discussed that it makes connections. So here you can see the actin filaments are both attached to itself with alpha actinin. Okay, then we have got the vinculin and talin, which is making interactions with the integrins, and where the integrins is in connections with the extracellular matrix. So this is how your focal adhesion is formed. Now coming to the hemidesmosomes. Now hemidesmosomes intermediate filaments आपके involved रहते हैं यहाँ पे. So you have got BP230, BP180 if you remember, right? So we have talked about this entire figure, same figure. So intermediate filaments and plectin, okay, they are in connections with each other with the help of the BP230 and 180, making interactions with the integrin molecule. Okay, now these integrin molecules are in cross connections with the laminin. As you can see, these are the laminin where it has got the binding area for the collagen, for the proteoglycan, right? So this is how your hemidesmosome is formed. Okay, and what is basal lamella? Basal lamella, which is made up of collagen, fibronectin, all those proteins, which is included. Okay, is this two junctions of cell and the matrix is clear, both of you? This is something which you have to remember also. Okay, so remember the figure, try to make it, and just make it in such a way that you remember, okay, alpha actinin is present between the actin filaments, and then they are in connections with the integrins with the help of the vinculin and the talon, and integrins are making connections with the extracellular matrix. And whenever cell is moving, these focal adhesions are formed and broken, so that the cells are able to move ahead. Okay, yeah. Any doubts in this figure? Okay. Okay, this is the small video. So these videos now, like it's directly from the Cooper itself. So whenever they talk about certain points, like about the focal adhesion ho gaya, yeah, anything else, they try to give the lab based, videos which has been conducted. Okay, so this is again in the human still the focal adhesion dynamics. So whatever is shining is the focal adhesion when a cell is moving. Is the screen visible to you?
Yes, ma'am.
Okay, so I don't think there is a volume in this, but when you see when a cell is moving, you can see something sparkling, this white color thing, right? So those are the focal adhesion which is formed and then again broken, formed and broken. Did you get it? It is very fast. Okay, so these white color things which you see as spikes, so those are the new focal adhesion forms, and then it gets broken because when a cell is moving, then it is forming a new focal adhesion here, and at the back it retracts. So there the focal adhesions are being broken. Got it?
Yes, ma'am.
Okay. Now this is another video. This will help you guys in understanding like what happens if that particular protein is mutated. Okay. So let's watch this one also. I don't think there is any volume. If integrin is blocked. So this is the antibody which is, bound to the integrin, because of which integrin is not able to make connections with the collagen fiber present in the extracellular matrix. Now myosin is being inactivated. What will happen? No connections with the actin. Integrins will not make any interactions with the extracellular matrix. Again. So talon is responsible making interactions between the actin and the integrin. If talon is not working, no connections. And eventually what we see is integrin is not able to make interactions with the extracellular matrix. Okay. So it means the cell won't be able to adhere to the extracellular matrix. Okay. जो एक सेल की प्रॉपर्टी होती है, like to be remain in to remain attached to their substratum, that will be loose. Okay. So hemidesmosomes mediate epithelial cell attachments at which a specific integrin known as alpha 6 beta 4 is linked to intermediate filaments instead of to the actin. Okay, so this integrin will also bind to laminin. So hemidesmosomes anchor epithelial cells to the basal lamina. So just now in the hemidesmosomes we saw here. So instead of actin, it is the intermediate filaments, and with the help of the laminin, okay, sorry, with the help of the alpha 6 and beta 4 integrin, they are interacting with the laminin present in the basal lamina. Okay, now coming to the animal cell growth. Just as plant cells modify their cell walls to grow, animal cells also modify the extracellular matrix as they grow and migrate. So in the case of the plants, what do we saw? That the cell wall, okay, they become loose with the help of the expansins, and the cell wall becomes loose and it gets stretched when the water is being stored in the vacuole, right? Now in the same way here, in the case of the animal cells, it secrete enzymes which digest glycosaminoglycans as well as a large family of proteases which are secreted by the cells. Okay, for example, collagenase. So this enzyme will digest variety of the proteins, collagens, laminins, which is present in the extracellular matrix, as well as the surface receptor and the addition molecules. जिससे क्या होगा, सारे connections आपके टूट जाएंगे with the extracellular matrix, and the collagenase will be degrading your collagen. As you can see here, it will be degrading your collagen into the collagen fragments. Okay, now if you remember, these collagen are nothing but your amino acids, right? If you remember the sequence also, glycine XY, where X can be many of the times X is proline and Y is your hydroxyproline. Remember, and it is a triple helical structure, right? So when collagenase is breaking your collagen into collagen fragments, eventually it is degraded into amino acids. Okay, so these amino acids can be taken for further growth of the cell, to make further proteins inside of the cell, या कोई अलग तरीके का receptor बनाना है. So depending on that, so nothing goes waste. Okay, so all these degraded products can be recycled. Now, the metalloproteases play important roles not only in the normal movements of the cells during development, but also in the growth and metastasis of the cancer. So we'll come to that. So see, guys, over here, what is happening? So it means what? तो cancer cells आपके बहुत fast movement करते हैं, ठीक है? So just imagine the amount of the metalloproteases they secrete out and the overexpression of the integrin molecules, you know, so when they are moving, so जो आपके focal adhesion जो बनते हैं, okay, so that is going to be like higher in number, so that the cancer cells can move faster, and the metalloproteases, so that they can just chop off the extracellular matrix and they can keep on adding. Okay, so for example, let's see here, you have got the blood vessels, and the blood vessels are made up of your endothelial cells. Okay, 1 second. So they are made up of your endothelial cells. Now these endothelial cells, what happens, ऐसे आपके present रहते हैं, right? Now in between of these endothelial cells, so as we have discussed, between these also the connections are there. Those are broken, okay, by the cell extracellular enzymes. They penetrate and they squeeze themselves, okay, and the correct word for this is called the extravasation. Okay, so they come out between the two cells, they start migrating. Now migrating means, as you can see here, they start clearing the area by secreting your MMP. What is MMP? Matrix metalloproteases. Okay, and the tissue destruction wherever it is getting localized in the case of the cancer cells. Now cancer cells also release VEGF, vascular and endothelial growth factor. So it is going to induce a formation of your new blood vessels so that the oxygen supply, so that all the glucose which is present over here can be given to the cancer cells. Okay, so angiogenesis means formation of new blood vessels. I think I have a video on this, like how the cancer cells are migrating. Okay, let's watch this.
Normal cells grow in a controlled manner to form tissues that form organs with specific functions. Malignant cells are defined by their ability to invade adjacent normal structures and be disseminated or metastasize. Malignant tumors can metastasize at any point. They do so by having cells break off from the main tumor, enter the bloodstream and/or lymphatic channels, and travel to other parts of the body to initiate a new tumor. Their ability to invade eventually affects the function of the normal tissue into which they are growing. Metastases is a multifactorial process involving complex interactions between tumor cells, vessels of the lymph and vascular systems, the basement membrane surrounding those vessels, and the extracellular matrix, which consists mainly of collagen. Let's take a closer look at how the EGFR pathway activates and modulates metastases. When the appropriate signals enter the cell, a complex chain of events within the cytoplasm is set in motion. These events eventually lead into the cell nucleus, where the transcription of genes regulating cell cycle progression and cell growth are stimulated. One protein produced through this cell activation process is the enzyme matrix metalloproteinase, or MMP. When a tumor cell metastasizes, it breaks off from the main tumor and enters the extracellular space. Tumor cells secrete MMP, which degrades the collagenous extracellular matrix, or ECM, breaking through the basement membrane that surround the tumor and allowing the tumor cell to migrate toward the blood or lymph vessels. When the MMPs reach the vessel, they break down the basement membrane surrounding the vessel through enzymatic action, opening access to the epithelial cells lining the vessel. Tumor cells can then migrate into the blood and lymph by entering through the tight junctions of the epithelial cells. The tumor cells are then transported through the blood and lymph to other tissues. It is known that metastatic tumor cells tend to target some organs more than others, although the reason why is poorly understood. The migration of tumor cells into the organs is very much like the recruitment of white blood cells to tissues after injury. Initially, there is weak adhesion of the tumor cells to endothelial cells, which allows the cell to roll along the vessel lining until stronger bonds are formed. Once the metastatic cells are securely attached to the endothelial lining, they leave the vessel and enter the tissue. They also leave an open pathway that allows less aggressive tumor cells to invade the tissue and grow.
Okay. So did you get it? Yeah. Did you get it? Like how the cancer cell is migrating? It is just that like normal cell also migrate in that way only. The overexpression of all these enzymatic enzymes and the cell surface receptor is more in the case of the cancer cells as compared to the normal cells. Okay, and the normal cells won't, it won't induce angiogenesis. Like in the case of the cancer cells, they need all the extra glucose, so that's why they will induce the angiogenesis. Did you get it? All the points?
Yes, ma'am.
Yeah. Okay. Now that was about the cell matrix. Now coming to the cell-cell interactions. Okay. So this is just, I'm going to just introduce about the cell interactions. 1 second. So as you can see here, there are mainly two types of interactions they have shown. Right? In between the cell, and there are three types of junctions which are playing important roles. Okay. So you might have heard about, see, first interaction is between the cell, right? And then between the cell and the matrix cell, right? So those are the different junctions which are formed. So in the upcoming lectures, we will be talking about the adherence junctions. Now, please guys, don't get confused. That time we talked about the adhesion proteins. Okay, SION, here adherence, ERENS. Then there is something called tight junctions, and then we have got the gap junctions. Okay, so this we will be doing in the upcoming lectures. Yeah. So from the next lecture onwards, we will be continuing with those adherence, tight, and the gap junctions. And yeah, so with that, the topic will be completed. Yeah, so any doubts would you like to ask? Okay.